All right, so this is the last step, walking through this first example of three-point mapping or three-point test cross analysis. Again, we're looking at this example where Rollins-Emerson crossed corn plants together, investigating three traits at the same time. And in the previous videos, we talked about analyzing the data to determine that indeed these three genes controlling these three traits are on the same chromosome. So we talked about that in video 1. Then in video 2, we talked about how to use the data of the cross to determine which alleles were together on the parental chromosomes. We call that the parental allele combinations. We figured that out. Then, also in video number 2, we determined which of the genes was in the middle, so we could determine the gene order on the chromosome. The last thing we want to determine using the data from this question is what are the distances between each of the gene pairs on that chromosome. So that's what we'll answer in this last video. So again, the last question we want to answer is what is the distance between each pair of genes on the chromosome? Again, we know we did this cross where we had this F1 progeny. One of the chromosomes in that F1 progeny had nothing but dominant alleles. The other chromosome had nothing but recessive alleles. We said those were the parental allele combinations. We again crossed to a homozygous recessive individual. Both chromosomes carried just the recessive alleles. And then we saw this outcome from the cross where the most common outcomes represented our parental allele combinations. And then we talked about using the least common outcomes to determine which gene was in the middle. We called those double crossovers. Okay. And so again, the last thing we want to do is use this data to figure out the distance between each gene pair. So again, we determined that the order of the genes was V, GL, VA. So we want to figure out three distances. What's the distance from V to GL? What's the distance from GL to VA? And then what is the distance from V to VA? So there are three pairs of genes, and we want to determine each of the distances. All right, so the way I start this part of the question is that I write down the parental allele combinations side by side. So again, in this F1 fly, in this F1 corn plant, one of the chromosomes has uppercase V, uppercase GL, uppercase VA right here. So I've written that down. And then right next to it, I'm going to write the other chromosome. I'm going to write it right below like this. And by having the two chromosomes written down with the genes in the correct order, I can sort of drag my finger across and figure out what outcomes do I get when crossing over happens in certain locations. All right? So the next thing I do is next to all of these phenotypic outcomes, I write which combination of alleles did we get from this first parent. Remember, we know what we got from the second parent. We always get recessive alleles. So what we see in the phenotype tells us which of the alleles we got from the first parent. Did we get a dominant allele for that trait or the recessive allele for that trait? So we talked through how to determine the genotype of the parents, the parental gene combinations. Again, this one has all the recessive traits, so it has all recessive alleles. Number 5 here has all dominant traits, so it has all dominant alleles. Then in the last video, we already talked about assigning the alleles for the double crossover progeny. This one has the recessive yellow, the dominant rough, and the recessive variable, whereas the other one had the dominant green, recessive glossy, and dominant normal. So we just keep filling this in. Yellow, rough, normal, your yellow, glossy, normal, well that's recessive, recessive, dominant. Yellow, rough, normal is recessive, dominant, dominant. Green, rough, variable is dominant, dominant, recessive. And green, glossy, variable is dominant, recessive, recessive. Okay. So now I can look at these allele combinations that I got from the first parent and think about how would you have to rearrange these alleles to get that outcome? So let's take this very first one as an example. How can I get a recessive V, a dominant GL, and a dominant VA? So I just go over here and I go, well, I start with recessive V. And as I drag my finger down the chromosome, at this point, before I hit the GL gene, I have to cross over to the other chromosome to pick up the dominant GL. And then if I stay over there, I get the dominant VA. So what I've just learned is to get this combination, lowercase v, uppercase GL, uppercase VA, I had to get a crossover event here between the V and the GL genes. So I write that down. I say, well, right here, this genotype represents a crossover between the genes V and the gene GL. And then I just keep going down the list like that. So what about if I wanted to have the recessive V, recessive GL, dominant VA? Again, start with the recessive V, keep the recessive GL, but then at this point I have to cross up to the other chromosome and get the dominant VA. So I got a crossing over between GL and VA. So for this one, I say the crossover occurred between GL and VA. All right, what about this third one here, number 3? This is one of the infrequent ones. This is what we call a double crossover. I'm going to get lowercase v, uppercase GL, lowercase VA. So again, if I drag my finger along, lowercase v, to get the uppercase GL, I have to cross over here between V and GL. But then to get the lowercase VA back, I have to cross over again between GL and VA. So I have a crossover event here between V and GL and a crossover event between GL and VA. So I say that this had crossover between V and GL as well as between GL and VA. This is what we call a double crossover progeny. Again, that's why it has such a low frequency. Again, these most common ones, they're the parental gene combinations. These have no crossing over occurring, right? We just kept the alleles in the same combination they already existed on the parental chromosomes. So these ones do not involve any crossing over. I'm just going to hide myself so we can see more data here. So if we skip the parents, then we get down here, uppercase V, lowercase GL, uppercase VA. Again, this is the infrequent one. This is a double crossover again. Uppercase V, lowercase GL, uppercase VA. I had to cross over between V and GL and between GL and VA. So double crossover, and I write down where the crossover events occurred. Then I look at the next piece of data. Big V, big GL, little VA. Big V, big GL, little VA. I have these two genes together, but then I pick up the third from the other chromosome. So we got crossing over between GL and VA. So I write that down. And then the last bit, big V, little GL, little VA. So big G, little GL, little VA. I got crossing over between the V and the GL genes. The crossover occurred between V and GL. So this is the first step in trying to answer this question. What is the distance between each of the genes? First, I need to recognize which of these outcomes has crossovers where, right? So now I know when there were crossing over between V and GL. This one, this one, this one, and that one. And when there was crossing over between GL and VA. Again, here, 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 and here. All right. So we're going to use that information to calculate the distances. So I'll just bring myself back. So how do we calculate the distances? Well, we just calculate the recombination frequency. In order to calculate the distance, we just take the number of recombinants between those genes and divide by the total number of progeny. So the total number of progeny is super easy. It's listed right there at the bottom. We looked at 726 total. All right, so we need to figure out what number to divide by 726 to get our answer. So let's look at each gene pair one at a time. So what about the V to GL distance? Okay, so I have to find the V to GL recombinants. Again, it's number 1 right here. It was number 8 at the very bottom. and it was number 6 and number 3 as well. So I'm going to take those numbers, 60, 62, 4, and 7. Those represent all the recombination events that occurred between the V gene and the GL gene. I add that up, divide by 726. If you do that math, you should get 0.183. So that's our recombination frequency, or we could say that it's 18.3 map units, or we could say that it's 18.3 centimorgans. So centimorgans and map units are common distance measurements in genetics, and that's what we'll most commonly use. So we just determined for the V to GL gene distance, it's 18.3 map units. All right, what about the GL to VA distance? Again, we just have to find the recombinants that occurred between those two genes. It's going to be number 2 right here. Again, number 3. We're going to keep number 6 and number 7. Those 4 offspring classes represent the recombinants between GL and VA. So we're going to take their frequencies, number 40, or 48 of them, plus 40, plus 4, plus 7. Divide by 726 again the total number of offspring. We get 0.136, or 13.6 map units, or 13.6 centimorgans. Okay. And then again, the last pair of genes is the outer pair, V to VA. V to VA represents the outside pair of genes. Every recombination took place in between those genes. So we're going to add up the 60, the 48, the 4, the 7, the 40, and the 62. But there's one specific detail you have to pay close attention to, and that's to think about these double recombinants. Remember, to get these, we had a recombination event between V and GL and a recombination event between GL and VA. That means that between V and VA, there were actually 2 recombination events. So we have to count those ones twice. So we're going to take every recombinant. So I've got 60 plus 62 plus 48 plus 40. And we're going to take the double recombinants twice. So I've got plus 4 plus 7, and then again plus 4 plus 7. So I add all that up, and then I divide by 726, and I get 0.319, or 31.9 map units, or 31.9 centimorgans. So, if I was answering a question, I would write down the data like this. We just determined that the distance from V to GL is 18.3 map units. The distance from GL to VA is 13.6 map units. And the distance from V to VA is 31.9 map units. And we're finally done. Those are all the questions we're going to answer in these 3-point test cross analysis kinds of questions. Is there evidence that the genes are on the same chromosome? What parental allele combinations are there? Which gene is in the middle? And then how far apart are each of the gene pairs? So I hope you'll use these videos, go back, pause, look at the numbers, think about how you do the calculations yourself, and use it to help you solve this kind of question. Again, these are the hardest questions that I seem to ask during the semester. This is where the most struggles are. So I hope you'll use this video to help, and that you'll bring your questions to class so we can talk it over. All right, I'll talk to you soon.